thanks everyone for showing up tonight and coming to hear the story about the Pacific Flyway and the book that we put out just recently on that very topic. So I think what I'll try to do is we'll see if we can get this all started for you so you can have a look at the nice pictures and so on. That's good. Okay, great. That's fantastic. All right, I want to bring you up to speed on some of the latest research that we've uncovered in the migration of birds. As we speak right now, the many birds are migrating north to in their breeding season, so that it's very timely that we should do this. And a few years ago, my co-authors, who you can see here on the screen, Audrey Benedict and Jeff Hammerson, approached me to collaborate on this book on what we've discovered. And we put together this book full of beautiful photographs and the story of migration. So this is really a story written by three of us, and brings you up to date on some of the latest findings in bird migrations. Now, when birds are migrating, of course, they're moving house and they're moving across the continent in huge numbers. And the way they fly and the routes that they take is what we're referring to as these flyways. And each of these photos that you see here, these are all in the book. Most of them are in the book. And you'll see down on the bottom, the photographers. We went through 8,000 photographs to choose the ones for the book. So it took a lot of work, a lot of effort, but some beautiful photographers out there. So many birds are doing this, moving house and moving from the south into the north to breed. And many of them are like the snowbirds here. They go south for the winter, get away from the snow, and move back north as the snow melts. And that's generally what we think about as birds moving north and south, but it's much more complex than that. We've discovered that birds have different flight plans, different species have different flight plans, and even different age classes have different flight plans. And as we discover more and more about these migrations, we're discovering that birds are, the migrations are very, very complex. We've learned a lot from birds, but the question now is, what can they teach us? What can we learn from this migration of birds that we didn't know before? Well, one of the first things that people who were studying birds came up with was this concept of flyways. This is the Pacific Flyway that I've illustrated here. But there's also an Atlantic flyway, there's a central flyway, and then there's several more around the world. There's about 8 major flyways. And the concept here, of course, is, as you can see, the birds fly south for the winter and fly north for the summer. But what we found was that really doesn't describe what's going on. These flyways, the concept of the flyways was really developed by hunters, duck hunters, in a way to manage movements of birds and to set regulations on how many birds could be taken. And so many of the ducks will make this kind of a migration, but many of the other birds on the flyway, as we've discovered, have quite a different concept of migration. So if we look at seabirds, for example, we have a lot of seabirds that migrate up and down the Pacific Flyway, up along our coast, and they're coming from all over the Pacific. They're not just going up and down, north and south, they're flying in all different sorts of directions. There are species like the puffins, the tufted puffin nests here off the British Columbia coast that appear to move mostly out into the sea and back for the breeding season. They're moving more east and west than north and south. There are gulls, such as the Heermann's gulls, that nest down on the islands in the Sea of Cortez, and in the late summer they come north. Instead of going south, they come north. They come here, we'll see them as far north as about Victoria, a few of them straggling into the southern Salish Sea. They're that beautiful slate-colored gull with the bright red bill. They hang around until about October or so, and then they head south again after being out for away from the breeding grounds through most of the summer. So their migration is in reverse to what we kind of think of, and then they catch on and they go south again. And then there's species like this, like the shearwaters, that do this flight all the way across the Pacific. And we have these, oops, let's go back to this one. We have these huge numbers of shearwaters going down our coast in the late summer, in August. And in fact, when we look at all the birds in British Columbia, the sooty shearwater is probably the most numerous. There are estimates of 25 million shearwaters that move up and down our coast. They breed in New Zealand, move up along the Asian coast, and then they come over through Alaska. And in late summer, they're coming down our coast, heading to California, and eventually will make their way back to New Zealand. So it's hardly a north-south migration. It's more like a circular kind of migration, leaving their breeding grounds down in off New Zealand and migrating north and then south, but along around the entire Pacific. Oops, kind of tough time with this one. Oh. We also have birds that come across to feed here along our coast, and some of the discoveries a few years ago of both the Laysands albatross and the black-footed albatross. They both breed in the islands in Hawaii. Black-footed albatross come over here and feed during the summer and carry the food back to their chicks on the Hawaiian Islands. So they cross halfway across the Pacific to get the food. And some of the other albatrosses will move even further than that, moving up into Alaska and back and going back and forth across the Pacific. So many seabirds can do this, make this kind of movement back and forth very, very easily. One of the reasons that they come here is that along our coast, we have what is called an upwelling zone through the summer. And the way it works is, if you look at the coast here, that when we get these high pressure systems, you'll see when the weather people are talking about good weather, and they say in the North Pacific, we have a high pressure system blowing. The winds are turning clockwise around the high like this. And what's doing is it's pulling the air offshore, off the British Columbia coast, and it has to be replaced. And what happens when that surface wind blows offshore is there's cool nutrient water from the depths that come up to replace it. And it comes up off the continental shelf, comes into the photic zone where the sunshine can reach it, and the plankton start to blossom, start to bloom. And we end up with these massive plankton blooms, starting with the phytoplankton, that's the plants, and then followed by the zooplankton. So that's all driven by these offshore winds. And you can actually see them if you go to the NASA website. You look, this is down in the Sea of Cortez, but all that green that you see there is plankton, and that's the plankton blooms that are being formed by this. And this lasts on our coast starting about now, believe it or not. We look out here today and we've got these cold weather fronts coming through, and it's just not really conducive. All through the winter, we have the low pressure systems, and that drives all this rain that we're still getting today onto the coast. But it won't be long, and it'll switch to the high pressure systems, and then we'll start to see all this upwelling and all the plankton. And as a result of that, these meadows become absolutely full of plankton. And this is the basis of the food chain, I think probably most of you know. So what that does is, with all the plankton, it attracts all these fish, and they're coming in here, the small fish, sardines, herring, anchovies, all kinds of small fish to feed on that. And of course, wherever there's lots of small fish, there's also lots of birds. And you can see right in the middle there, there's a sea lion, and they're pursuing the fish. And so that's how it works. That's how this all works through the winter. These switching from the low pressure in the winter to the high pressure, it creates that conveyor belt of food along the coast that attracts all these birds. And that's why they come here. They migrate here to breed, and they leave in the fall when all of this closes down. Now, I mentioned at the beginning that the flyway was really based on waterfowl, and it was largely waterfowl in North America. But now we know that actually some of these waterfowl species can migrate as far as South America. And we also know that some go as far as Siberia. So some of the teal, cinnamon teal, and so on will be down in South America. And I think probably most of you know many of our, well, all of our snow geese that go through the Salish Sea end up out on Wrangel Island in Siberia. And so that's their migration route. But there's lots of other species as well, including the brant, which are now in the Salish Sea. And these are ones that feed on eelgrass. So they're along all the beaches that have eelgrass, feeding on that, putting on the fuel to be able to make the migration to the next stopover somewhere in Alaska, and eventually up onto the breeding grounds. And so these birds are quite remarkable in that they move north along the coast, When they come south, they take advantage of those winds I was just telling you about, the high pressure winds. They catch those and they can fly all the way from Alaska right to Mexico nonstop. It takes them about 60 hours to do it, but they're reliant on those winds. And you'll see through this story that the winds play a really vital role on many of these species on how they migrate. Another one, which is very numerous here in migration, is the pintail, the northern pintail. And you can see how they got their name with that long pintail. Many of these will migrate down into California. We have lots here in the winter as well, but large numbers down in California. And we used to think that when they migrate, they followed these stopover sites north like a set of stepping stones, as if they were flying from airport to airport as they went north. So places like the Fraser River Delta and the Stickeen Delta and so on, we thought were probably critical stopovers for them. Well, since then, they put some transmitters on them, and some of the birds do that. But they found that some of these pintail are capable of flying nonstop from California right up into the Arctic, where they breed. So it really changed our view of this, just what these animals are capable of. Another interesting one is the eared grebes. We have eared grebes breeding in some of our interior lakes. They gather on huge numbers down on the Salted Sea and Mono Lake, and then eventually finish their migration down into the Sea of Cortez. And as you can see, they're all jammed together in this this photo. Sean Boyd, who has been studying these for the Canadian Wildlife Service, has counted the numbers, huge numbers in these two lakes, Mono Lake and the Salton Sea. And it's one of the things as conservationists that really concern us, that when you get a huge population from a large area, all concentrated in one spot, they're very vulnerable to any kind of a catastrophe in that one location. And this is the case for many migratory birds. It's that they might winter across a wide area, and they might breed across a wide area, but on migration, they concentrate in those airports, and it's so important that those places be protected. Now, I've talked about some of these species that are moving north and south, but we also have some that go east and west. So we have loons out here on the coast now, and they're getting ready to migrate, and they will migrate. Some of them will go a little bit north, but many of them will just go into the interior of the province and maybe up into Alberta. Our western grebes that are here in the Salish Sea will migrate as far east as Manitoba. So they have an east-west migration. And we see that among some of the gulls, the California gull and ring-billed gull will do a migration like that. So it's really changed our view. The flyway is not a north-south migration at all. It's very much a east-west, north-south, just about any direction you can think of. And even sandhill cranes, the huge numbers in California coming up through our interior, and we don't get huge numbers of these through the Salish Sea, but there are good numbers that migrate up through the prairies and in the interior of British Columbia. And one of them that you can see right now in the Salish Sea in huge numbers is this. This is the surf scoter. I think you're probably all familiar with it. The surf scoter arrives here usually in about October, November, stays for the winter. And the males are the black birds, back to the head and orange bill, and the females are the more muted brown colored birds. They migrate north at this time of year and are in very, very large numbers wherever there is herring. They're after the herring spawn, and they're after the eggs. So this is a herring spawn taking place up the coast. You can see the white in the water is all the sperm from the male herring that they release into the water. Females enter their lady eggs, and the eggs become fertilized, and then after about 14 days or so, they hatch out into small herring and then move out into the deep water. So what the scoters are after are the eggs. There are other species that will come for the fish, but in the case of the scoters, they're largely coming to feed on the eggs. And those ones from some of the radio transmitters that we put on come from as far as California and come up here for this very reason. And there seems to be, it seems plausible that they actually time the migration to coincide with the spawn as it moves north. And some of my colleagues have called it the silver wave as it moves north, following the herring as they breed further and further north as the season progresses. So here this is one of Leon Gertzman's. You probably know Leon, who lives here in Vancouver. One of his photos of all the male surf scoters coming in to dive. Well, there's some females there too, diving down to get eggs and get ready for migration. And the other group that I want to just tell you about, and one that we've done a lot of work on, is the shorebirds. There's about 50 species of shorebirds that migrate through the Salish Sea, and a few of them are very numerous, and most of them are in fairly much smaller numbers. One of the most numerous ones is this little guy. This is the western sandpiper. And in the next about 10 days, maybe 2 weeks, huge numbers are going to come through the Fraser River Delta. And if you haven't seen this, and you have an opportunity, if you live on this side, try to get out to Brunswick Point, which is just right near the Rifle Bird Sanctuary. And if you get out there on the rising tide, you could see 100,000 or more of these sandpipers. And they create these huge flocks, wheeling flocks. They're migrating all the way from South America, some of them as far as Chile, but more like Ecuador. Large numbers are in Panama. There's some in Mexico. There's probably about 3 to 4 million of them. And they'll stream up the coast through mostly from about mid-April, starting about now, until about mid-May. The peak is right at the end of April, and it's a real sight to behold. There are many other species like the godwits and things that are also coming through, marble godwits. We don't have them here, but in coming up the coast in other locations, you get that species as well. So where are they going? These sandpipers, many of them are going up to the Yukon-Kuskokwim Delta. And this is a shorebird Eden. It's located in the mouths of the Yukon and the Kuskokwim River on the west side of Alaska. And the Americans have set this aside as a wildlife refuge. And you can see it's just an ideal spot for shorebirds. All the wetlands full of insects, nice little dry hummocks and things where they can nest. And so this is probably the most important shorebird habitat, breeding habitat for shorebirds in the world. And this is where our western sandpipers go. So how do they do it? How do they make this migration? Well, one of the things we discovered early on was the birds, as I mentioned to you about earlier, follow the winds. And we discovered that when using transmitters and a bunch of other kind of analyses, we discovered that the sandpipers, it looked like they really relied on these winds to make the migration. So let's go back and think about this thing with the low and high pressure system. Now, when we have a low pressure system like we have today with the winds circulating counterclockwise around the low, these winds are coming from the south and coming up around the low pressure system along the coast. And it's exactly what the sandpipers need to migrate. They go at high elevation, so they're up over 1,000 meters, and they catch the high elevation winds, make the flights through. And we did a little analysis of this and looked at how they did it. And it was really apparent that they needed these winds to make the migration to arrive fat enough to breed up in Alaska. So when they're flying, the other discovery that has just come out in the last 10 years or less is that when they land on these mud flats, we wondered for the longest time what they were eating. They seemed to be just probing around and getting nothing. But Bob Elner at the Canadian Wildlife Service and Tomohiro Kawai from Japan and a few others discovered that they were feeding on what is called biofilm on the surface of the mud. And the biofilm is a combination of diatoms and bacteria and a few other things that produce a sort of a carbohydrate along on the surface. And what these sandpipers do is they have a special tongue and a special bill for picking up these blobs of mud. And about 50% of the fat that they put on could be coming from biofilm. Since they discovered it here on the Fraser Delta, they found that this happens among other species in other places of the world. And this has become actually a whole new food web in the estuaries. So this was discovered right here in the Salish Sea on the Fraser River Delta. So biofilm is one of the major sources of fuel for them, which they put on. Now, the sandpipers, this is them on migration. You can see the bright rusty red color, ready to migrate. They're just like a little butterball when they put on all this fat, and they're one of the favorite foods of the peregrine falcon. So the other side of this story is what is the role of the peregrine falcon in all of this? As many of you probably know, the peregrine falcons around the world suffered, as many birds of prey did, from DDT contamination back in the 60s and 70s and through the 80s and so on. Fortunately, now, with the ban on all of this, most of the birds of prey are back to their former numbers. So that's a conservation success story. But for many years, the shorebirds got away with this. They didn't have the predators around, and they could just migrate north and south. Life was pretty good for them. But with the return of the peregrine falcon, all of a sudden it's changed the whole picture. And we now are looking at this, is that these predators, they're timing their migration, of course, to catch these sandpipers when they're fat. And we're finding that the birds are changing their strategy too. It's like some sort of a computer game going on here. So think of it as a sandpiper. It's feeding. It's got to put on a certain amount of fat. It's got to carry that as fuel to get to the next stopover site. But if it puts on too much fat, it's going to get caught by one of these peregrine falcons. So how much do you put on, and how should you do this? Well, it kind of depends where you are in the migration. If you've got lots of time and you can sort of goof around a bit, just avoid the places where there's peregrines. And this is exactly what we found, is that you can see sandpipers if you go to Boundary Bay in the summertime. You watch where the sandpipers are. They'll come up towards the dike, and at about 300 meters, maybe a little less, they stop. And the reason for that is that as an approaching peregrine falcon flying fast but low over the marsh as a surprise attack, if the sandpiper sees it at about 200 meters, it gives the sandpiper enough time to get up and accelerate and get away from the peregrine falcon. If it gets any closer than that, then it's vulnerable. So what you'd expect then is the ones that would be come in closer will be the lean ones, the ones that really need the food, because there's lots of food up along the edge of the marsh. They are lean, they're fast, and they can come in a little bit closer. But as they get fatter, they should stay further offshore. And my former student, Andrea Pomeroy, studied this and found exactly that's what happens. As a sandpiper gets fatter, it stays further and further offshore where it's safer, because it's got the fat. It doesn't need to worry quite as much. It's the lean ones that come in. So you can see how sensitive they are to this. They can change all of this. We're finding this all over the world now, that these changes are brought on by these birds of prey. And it's like a game going on between the two of them. So the peregrine will try to adjust to that, and it may change its prey and all kinds of things. So there's all kinds of adjustments, and I can talk more and more about this later, but I just wanted to give you a taste of this, the role of the predators and how they are changing the world and the migration routes, the timing, the locations, and all of those things up and down our coast. I mentioned about these stopover sites being the airports. These are our snow geese up in Wrangel Island, and that's their breeding ground, and that's obviously to start the first airport that they're going to be on. And then as they come south, they'll be stopping on a variety of other places, ending up down on the Fraser Delta, a few of them going on down into California, but many of them staying here and moving back and forth between Skagit and the Fraser, depending on where they want to be. But there are all kinds of other airports for these birds. And in the book, you'll see this. We've listed all the major stopover sites up and down the coast. These are the branch up in Eisenbeck Lagoon that I mentioned to you before, that catch those winds and fly all the way across the Pacific to Mexico. Here they are putting on the fat out on eating eelgrass out in Eisenbeck Lagoon. And so you'll see photos through the entire book on these locations, and it tells you where they are, the kinds of birds, and more things about them. If you're interested in the flyway, how these birds make this migration, you can find it all in the book there. You can see they're beautiful, beautiful locations. Now, with all this information now, that we've learned, and what we can ask ourselves, what does this really teach us? And here in Canada, the loon plays a very important role for us. It's one of those species that's iconic for the country. We now know a lot about its migration. We know where they breed. We know some of the problems they're facing, and we can really get down to some conservation work. One of them here in the Salish Sea is to make sure we maintain those coastal areas with small fish that they depend on. And you can get involved in some of the bird counts and participate in that to see how they're doing, track them, see how the numbers are doing. They're kind of the barometer to this, telling us how lakes are doing in the interior where they breed, as well as how they're doing out here on the coast. And the movement of birds, cranes and ducks and geese and all the rest of it all are sending these signals, telling us more and more about the world that we live in. But I think one of the things that we can do, and what I'm so happy to see with transitions there in the programs you're doing, is informing people about what we have around us. And the next step for us is to think about this as part of our culture, part of this nature culture. The First Nations people here know about this in spades, and they've been, of course, their culture is deeply rooted in the landscape and in the Salish Sea. They have all kinds of stories and iconic images and so on that are all linked to it. But what the rest of us need to do is learn from this and try to build more on it. A few years ago, we did a film for the Knowledge Network where we went out looking for this, trying to find where people were actually doing this. And of course, we found it in the First Nations folks. But for the rest of us, there's little pieces right here and there. What we need to do is to tie our music and our art, our language, our food, our stories, all of these things to the Salish Sea. And we need to use the Salish Sea as the basis of our culture and how we're rooted here. So it's important to be telling stories like what I'm telling you tonight, but it's also important if you're interested in maybe in music or in art or in food or maybe in dance or whatever it is you're interested in, is try to tie it to the land and try to tie it to the Salish Sea to bring other people in, make it fun. And all of this we need to guide with science and be inspired by nature. But this is really a key element, I think, in our success is going to be see how we take it to the next step so that these things become part of the culture, and then you will want to conserve it because it's who we are. So when we ask this question, where will the birds take us next? I think that's where they will take us. I think we will see that when we start to connect with our culture here in around the Salish Sea and making it stronger and more embedded in nature, the birds will also show you that people elsewhere are doing the same thing with the same birds, and they are developing, cultures and responses to nature. And wouldn't it be a wonderful world to be able to see this and be able to have people share them, have people from other parts of the flyway come north and be part of the celebration that we have here, and we go there and let the birds do this. One of the senators that I worked with over in Asia said that, the birds are really a symbol of peace, because people, regardless of your politics, regardless of what, your religion, your background, whatever it is, if we study birds, we can all do this together. We can all study these birds. We get to know each other better. The birds are migrating and we're interested in how they're doing, where they're going, and if they're successful or not. And we're all interested in this. So let's do that, and the rest of the things will follow behind that. So with that, I'd like to close and just thank all the folks here. These are the acknowledgements of all the people that have been involved, or many of the people in this production.
I just want to say a big commend Rob on his presentation. That was amazing, and those photos were just beautiful. So I'm sorry that I won't be able to bring that level of photography to my presentation, but I know what we need to work on, I guess. Before I start, I just want to acknowledge that I am coming to you all today from the traditional territory of the Snuneymuxw First Nation. And I'm very excited to be here. As Simran said, my name is Alanna Vivani, and I work for the Mount Arrowsmith Biosphere Region Research Institute. Thank you for having me at Wonders of the Salish Sea and for Transition Salt Spring to be hosting this year. I'm very happy to be a part of it. So I'm just going to share my screen now. And if someone is able to let me know that we can see the presentation okay, I can get started.
Looks great.
Awesome. Okay. So just a little overview of some photos that we have here on the main page. On the left is a group of our volunteers who are processing some samples. The large one in the middle is just conducting some sampling. And the small triangle on the right shows a couple of eggs that we have found in some of our samples. To provide you with a brief overview of what to expect this evening, I'm going to give some background on our research institute, dive into Forage Fish of BC and the importance of monitoring, a look into our monitoring program and the citizen scientists who make it happen, the methodology that we use, some of our results, how we display those results. And then lastly, I'm going to touch on a spring forum that BC is hosting very shortly. So let's start with the Institute and the area that we are researching in. MABRI was established in 2014 at Vancouver Island University, and it's essentially the engine behind the Mount Arrowsmith Biosphere Region's research and educational programs. The Mount Arrowsmith Biosphere Region is a UNESCO designated biosphere that spans from Nanoose Bay north to Qualicum Bay. And we're dedicated to community engagement, relationship building, and knowledge sharing. So MABRI, the Research Institute, focuses on research and community engagement primarily within that UNESCO designated area, but we also uphold VIU's academic research plan by working on projects across Vancouver Island. So this yellow shaded area demonstrates the MABR, but as you'll see in some upcoming maps, the Forage Fish program has expanded beyond these boundaries for MABRI, but even further for the BC network. MABRI provides VIU undergrads and graduate students with hands-on experience in the field, and we are able to tackle a diverse set of projects through interdisciplinary work. So Forage Fish is just a small portion of the work that we do, and it just falls under our environmental branch of research. Now, to dive right into Forage Fish, they are these small, abundant schooling fishes that are a critical component to the marine ecosystem and the fisheries. They're a key prey item for larger predatory fish, marine mammals, and seabirds. Pacific sandlance alone are prey to about 45 species of sport and commercial fishes, 40 seabirds, and 12 marine mammal species. Specifically, humpback whales, minke whales, and harbor seals all depend on forage fish for about 75% of their diet. These fish are grouped based off of their ecological role rather than their taxonomy. So something a little bit different than what people are normally used to. Now, the role that they play, they're an integral part of the marine food web as they influence both predator and prey populations. They are the linkage between the lower and the higher trophic levels while they transfer energy throughout the entire food web. And there's evidence that dependent predator populations mirror the forage fish population fluctuations, so increasing and decreasing as a result of resource availability. And in 2009, a study was conducted that showed approximately 34% of coho salmon diet consisted of forage fish. On northern Vancouver Island, Pacific sandlance consists of 65% of the diet of adult Chinook salmon, which we know to be a key prey item for resident killer whales specifically. Now in BC, there are about 7 species of forage fish that reside along the coast, including Pacific herring, Pacific sardine, northern anchovy, eulachon, surf smelt, capelin, and Pacific sandlance. Each of these species have different geographic ranges that they frequent, different ecology, and different life cycles. For the purpose of MABRI's research, we are specifically focused on surf smelt and Pacific sandlance, as their spawning habitat is similar and easily accessible for sampling without requiring the need of a watercraft. Now, talking about their habitat use, forage fish and their spawning habitats are protected under Section 35 of the Federal Fisheries Act. However, at the time of the creation for this document, there was minimal data that was available regarding the location of forage fish spawning habitat along the coast of British Columbia. So generally, Pacific sandlance and surf smelt use the upper intertidal zone of beaches on Vancouver Island for spawning and embryonic development, where the sediment is composed of a sand or a mixture of sand, pea, and pebble gravel. While most people are really used to and familiar with herring spawning further down in the intertidal zone on eelgrass blades, Pacific sandlance and surf smelt eggs are dispersed in a zone that is fully uncovered during a low tide event. So in this photo here, you'll notice that this upper zone in the intertidal is where we'll find Pacific sandlance and surf smelt eggs. Now, this photo is of a beach station in Nanaimo called Morningside. It's actually a positive station for us where we have seen eggs from Pacific sandlance for the past 4 winters. The measuring tape down the center of the photo shows you the midline of where we would collect our samples. So sediment from above that tape to the left and below that tape to the right will be collected. And this whole sample area is just lower than that rack line, where you can see the driftwood has been moved up the shore and there's a bit of seaweed left there too. So we're focusing on that lower elevation where at a high tide event, this would have been covered by water. Now, due to the set of conditions required for suitable forage fish spawning habitat, the forage fish are exposed to several stressors. Literature has outlined that overhanging vegetation is a beneficial feature as it keeps the sediment beneath it cool and moist. And you can imagine this is especially important for summer spawning activity when there is a lot of sunlight available all day and it's just baking the sand. Lots of trees and vegetation from development can pose negative effects on this. Similarly, increasing temperatures due to climate change can also play a role in the lower survivability due to extending beyond thermal tolerance ranges and contributing to desiccation. As they require suitable sediment to allow their eggs to attach to for sufficient oxygenation, the construction of seawall armoring, such as large boulders and stones or other riprap structures, can decrease the suitable sediment by having water hit the armoring and compact the sediment so much that the eggs are not able to either penetrate the sand or there's not enough oxygen for the eggs to be viable. And of course, these species have also been susceptible to overfishing. So some other stressors that we can include would be seaweed harvesting. Seaweed will oftentimes provide a moist environment underneath it to have the eggs stay viable, and also just pollution from stormwater and any other sources that may be polluting the beaches. Now we're going to talk a little bit about Pacific sandlance and next surf smelt, as those are our 2 main species that we're looking at. Adult Pacific sandlance are silver, they are elongated and narrow, and they grow up to about 20 centimeters long, living anywhere between 5 and 7 years. Sometimes people recognize Pacific sandlance more as being called needlefish or needle nose fish, so might be something you're more familiar with. Their distribution spans the length of the Pacific Northwest coastline and across the sea to Japan. Pacific sandlance spend days feeding in the water column and their nights buried in sandy sediment in the intertidal or subtidal regions in order to avoid predation. And from November to about mid-February, Pacific sandlance will choose sandy beaches or a mix of gravel pebbled beaches with sand to spawn on, targeting that high tide line to the mid-intertidal zone. And they haphazardly spawn, releasing their eggs, which are about 0.6 to 0.8 millimeters in diameter. And those eggs are vulnerable to dispersal by wind and wave action. Each egg attaches to multiple sand grains and will incubate for approximately 4 weeks. Now, surf smelt is another silvery colored fish that grows 20 to 25 centimeters in length and is found from California to Alaska as well. They are sexually dimorphic, which has the males showing a brown dorsal side and a yellow ventral side, when the females have a green dorsal side and more of a white ventral side. Surf smelt spawn year-round, with peak spawning seasons varying regionally, but literature in Washington has noted that most commonly spawning is occurring from June to September. However, our partners in Victoria have been seeing some spawning of surf smelt from January to March. They prefer beaches with small gravel, and they choose to broadcast spawn near the high tide line. Regardless of the season, surf smelt spawning has been found to be linked to tidal and lunar cycles, having the greatest abundance of spawning individuals present on high evening tides accompanied by a full moon. So soon after fertilization, a small distinctive fold in the egg's membrane forms, and it attaches to substrate in one location on the egg. They'll incubate for about 2 to 4 weeks. So why are we monitoring beaches in BC? Why are we focusing on these two species and doing forage fish research? The reason behind it is the data that we can retrieve from this will enable evidence-based decisions to be made. And some of these decisions are modifications to policies and management plans that can help protect their spawning beaches. So the hope is that beaches that they use will have the opportunity to maintain populations of forage fish to healthy levels. Washington Department of Fish and Wildlife began monitoring forage fish species in the early 70s, and systematic surveys of spawning habitat began in the late 80s. So they have this really extensive database identifying where and when Pacific sandlands and surf smelt have been spawning throughout Puget Sound. And they can use this information to protect intertidal regions where spawning activity is recorded. So think of it as how some beaches are closed to dogs in certain times of the year to help with the migratory bird populations. Now, BC's knowledge regarding windows and preferential site characteristics for spawning has been adopted from work completed in Washington state. So a lot of the information that we have, and when I mentioned that literature states this, that's provided from research done by Washington. MABRI, along with other organizations within BC, collectively known as the BC Forage Fish Network, are working towards closing data gaps and developing baseline data for forage fish spawning. To protect crucial habitat and spawning habitat, identification of habitat is really key. In British Columbia, very limited identification of spawning beaches is sort of there. We are about 4 decades behind Puget Sound surveying. And in 2005, the BC Shore Spawners Alliance, which was advocated by Ramona DeGraff, started to conduct beach surveys throughout the Strait of Georgia. She conducted extensive ground truthing during this time and did a lot of spearheading the movement to bring forage fish research to British Columbia. From 2013 to 2018, the island's trust fund completed suitable habitat spawning assessments for Pacific sandlands and surf smelt in the Gulf Islands, and added possible suitable spawning habitat sites to their MapIT site. Spawning beach data is still limited and somewhat sporadic for the BC Salish Sea due to past inconsistent data management. But for this reason, many organizations along coastal British Columbia have come together, forming that network I mentioned, to work on minimizing those gaps and build a strong database of monitoring efforts and results. So this started quite a few years ago, and we're still working to this day together. Now, the BC Forage Fish Monitoring Network is made up by a number of organizations. Our collaboration with the network has provided us with really strong partnerships all along BC's coast, especially in the Salish Sea, including but not limited to Project Watershed, Tsleil-Waututh Nation, Peninsula Streams and Shorelines, Loon Foundation, Sunshine Coast, Friends of Forage Fish, Howe Sound Biosphere Region Initiative, and Friends of Semiahmoo Bay. Like I said, we're working really hard to minimize the data gaps and ensure that we're covering as much geographical area as possible. And many of these organizations host their own network of citizen scientists who conduct a bulk of the sampling and processing, and then provide samples to be analyzed by the host network. Our Forage Fish monitoring program has 5 key components to it over the last few years, and then we have a 6th one that we're working towards for the future. So in 2017, our researchers were trained by Philip Dion from Washington Department of Fish and Wildlife on forage fish sampling methods that they developed over the past few years. Following the training session, we worked with a retired marine biologist to modify the sampling methods to better suit our program needs. The adjustments were really made because our study didn't focus on the density of spawns, but rather where the spawn was occurring and, of course, when it was occurring. We were looking to find beaches that could support forage fish spawning, what we refer to as positive beaches. Some of the notable changes that we made were utilizing larger containers to collect samples, so we had a better chance of finding eggs. We switched to the Wentworth sediment scale. We conducted bias sampling, really trying to select locations on a beach that looked best. And we included more environmental and beach station characteristics, such as aspect of the beach, any episodic or weather events, and the slope of the beach. Now from 2017 until now, Mabry has been sampling beaches along the eastern coastline of Vancouver Island. Initially, our beach stations were established and sampling efforts were focused around the Parksville-Qualicum Beach Wildlife Management Area. But since then, we have greatly expanded beyond the boundaries of the MABR. In the summer of 2018, our research assistants worked on identifying new sample locations along the coast, building and expanding our sampling area. Currently, there's anywhere from 30 to 90 beach stations that are sampled monthly with the help of our dedicated volunteer network. And this number really does fluctuate based on the presence of suitable sediment at the time of the visit, as well as volunteer capacity. And with some of the Indigenous knowledge that our partners have received and our citizen scientists have received, some of the groups are focusing their sampling efforts only during the winter season of Pacific sandland spawning, because surf smelt is not known to frequent their areas. In the past, we've also incorporated eDNA sampling for a couple of the winter seasons, and this was included to see how that sampling method can aid in identifying beaches that forage fish are spawning on. So eDNA refers to environmental DNA. It's collecting sediment samples from the same area that we were due our physical samples from, and trying to see if there's any surf smelt, any Pacific sandlands DNA that was left behind. And we would compare the results of the eDNA to the results of our microscopic analysis to see if we're finding the same things. If we're finding eDNA traces, are we also finding eggs? In 2018, we had a practicum student create predictive maps of forage fish spawning habitat using data collected from ShoreZone. WWF Canada collaborated with Coastal and Ocean Resources to develop a predictive mapping tool that could identify potential forage fish spawning beaches within the Salish Sea. And the ShoreZone data was compared to known forage fish beaches to determine attributes that are associated with spawning beaches. And this really helped further develop the predictive mapping tool. Some of the attributes that were used consisted of protected or semi-protected wave exposure, the beach type, beach composition in the upper intertidal region, dominant substrate, and any eelgrass biobands in the subtidal region. Map outputs were produced like this one that you see there, and they ran codes from established predictive tables, helping to identify possible spawning habitat. Our team used these then to ground truth as many of these suitable beaches as possible so that we can add some sites to our plethora of beach stations already. Since 2018, we've been hosting training sessions with community members. This happened once we were familiar with our sampling and with our methods. We wanted to expand the program and include citizen scientists. We worked to train groups to collect and process samples within their designated areas. And thanks to funding from our partners, we were able to equip these citizen scientists with their own sampling kits and their own processing kits. Initially, this training began on Gabriola Island with the Gabriola Island Shorekeepers Association and Thetis Island Nature Conservancy. And we worked really closely with these groups. It allowed us to understand the difficulties around having a citizen science group and also the necessary changes so that we could have really efficient sampling. Since then, we have added numerous groups and also some individuals, and we're constantly looking to expand not only our geographical range, but our network range too. We are continuing to reinforce the partnerships that we've made over the last couple of years. The creation of the BC Forage Fish Network really helped us, provided us the opportunity to not only share resources and validate results, but collaborate on future initiatives surrounding forage fish. And hosting refresher sessions for our citizen scientist groups allows us to not only recruit new volunteers, but ensure that there's no bad habits that are adopted. We also produce a bi-monthly newsletter to keep citizen scientists engaged and involved in the research. Now, looking forward for us, Mabry will be collaborating with our partners in the network to work on restoration on select beach stations. So this can range from soft shore work to beach nourishment as well. So a little short overview so far. Mabry has been involved with monitoring since 2017. We have established 117 different beach stations from Deep Bay to Cowichan Bay. This also includes Gabriola, Thetis, Pender, Saturna, and Salt Spring Island. And as of March 31st of this year, we, alongside our citizen scientists, have collected almost 1,800 samples. To date, we have 37 beach stations, which are on 31 beaches that have had egg identifications. And these are all Pacific Sandlands eggs. So our network has yet to find surf smelt, but we've been really, really fortunate in finding lots of Pacific Sandlands beaches. Now, the map on the left will show you our sample area, most of the sample area. And the yellow squares are indicative of sample sites that are adopted by our citizen scientists. The blue ones are ones that our team visits on our own. So there are about 117. What you can't see right now are the sites on Salt Spring, Pender, and Saturna, as they're off of the map there. But of the 117 sites, 78 of those are assigned to citizen scientists. So it's really without their involvement and without their dedication, we would not have been able to expand the program to how big it is now geographically. And we are continuing looking for new partnerships and more volunteers who want to get involved, because this work is just always expanding, and it's always great to keep building that database of sampling efforts. Now, the data collection methods were slightly modified for citizen scientists, primarily to reduce the amount of equipment that was necessary, but ensuring that the most important information is still being collected. Each group varies in size and how they undertake the sampling. Some groups will collect all the samples and process all of their samples as one group. Others will split into pairs and work in smaller teams to sample and process. But generally, the sampling teams will choose beaches that are close to them for ease of access and also fostering that sense of connection to the environment. Mabry's role in coordination involves providing support to volunteers for questions they may have, providing equipment and maintenance, supplying sampling schedules, hosting training and refresher events, conducting the analysis, of course, sharing results, and completing the data entry. We currently have about 10 citizen science groups who have trained with us and continue to work with us. You can see some of their logos there on the right-hand side. Now, I want to remind everyone how impactful this research has been in BC so far and showcase some of the great work that's being done outside of Mabry's region. This is only a couple of examples of partners who are contributing to Forage Fish research in British Columbia. There are many other organizations, as I had mentioned. First, I'll talk about Project Watershed. They operate just north of us in the Comox region and beyond. From November 2022 to February 2023 alone, their volunteers have registered just around 350 hours of work dedicated specifically to the Forage Fish project. So there's a lot of time involved in that. And they have a really, really amazing team that operates within the Courtenay-Comox region, but also on some of the neighboring islands. Now, to the right there, I have Peninsula Streams, and they operate south of us in the CRD region. They have approximately 58 active volunteers, and they have completed 842 surveys as of mid-March, and this is from the beginning of their program. They've had a total of 175 positive detections. 54 of those were in 2022, and 29 of those have already been done in 2023. So they're another power group there, just with their volunteers and really getting a lot of sampling done in and around Victoria and the Saanich Inlet. So we're going to go through the methodology right now of conducting some forage fish monitoring. The first is sample collection. So samples are collected only when a site has suitable sediment, and this will vary depending on, on the season. When we are focused on Pacific sandlands, we look for either straight sandy beaches or a mix of pea gravel that has a sand base. When looking for surf smelt, we look for a pea gravel that's mixed with pebble gravel and also has a sand base. And I want to emphasize when I say a sand base, it's really important that there's sand mixed in with the sediment, as it's the small sand grains that will attach to the eggs and keep them locked into the sediment there. So scoops of sediment are collected along a 30-meter measuring tape into a 4-liter container at that optimal tide height. Various beach characteristics are recorded to determine if there are any correlations to spawning activity. Characteristics and the photos are all sent to Mabry, where we conduct the data entry. Our next step is sample processing, and this happens in 2 steps, actually. So the first is sieving. The collected sediment sample is transferred to a collection of sieves and processed down to obtain an amount of sediment that the embryos would attach to. So we sieve the samples through a 4, a 2, and a 0.5 millimeter sieve, and we keep whatever is left remaining in that 0.5 millimeter sieve. As I mentioned, the eggs are around 1 millimeter in diameter, so they won't fall through that sieve, and they'll just be caught on top of it with any sand grains that are attached. That collection of sediment is placed into a holding container. The sieving allows us to focus our efforts on the sediment that has the highest probability of containing eggs. The next step in sample processing is vortexing. The sediment that we collected in that smallest sieve is then processed through a vortex machine, which you can see in the left side photo at the bottom there. The vortex machine is the newest and most efficient way of processing samples. This was developed by Washington Department of Fish and Wildlife. The recirculating system pumps water from a tote, as you can see in that photo, using a bilge pump that is run off of a marine battery, and it gets pumped into this blue bowl that's on top. The blue bowl is a piece of equipment used in gold panning. It has a raised center with a hole in the middle, and that allows the water that's vortexed to fall through that middle. The movement of the water creates a pressure gradient with lower pressure occurring in the center. And this lower pressure system allows the lighter material, such as the organics, to be lifted through that water column and then dropped through the center hole where it is caught in a 0.5 millimeter sieve below. We use a spatula to agitate the system as it is circulating, so that we can ensure that any eggs that may be buried under the sand have a chance to be lifted through the water column. The elevated hole in the center reduces the amount of sand that leaves the bowl, and that reduces the amount of sediment that later needs to be analyzed under the dissecting scope. Once the processing is complete, the sediment remaining on the sieve is transferred to a sample jar, and the initial large 4-liter sample collection fits into a small sample jar, which may only have a couple inches of sediment in it, as you can see in that middle photo. After processing, we get to work on analysis. Mabry at the moment conducts all analysis on samples that are collected by our citizen scientists and by our own team. We use a dissecting scope, and a teaspoon amount of sediment at a time is looked at thoroughly to find any eggs that it might contain. The number of eggs within a sample and the species type is recorded for every sample, and any eggs that are found are confirmed by experts in the field. After the sample analysis is complete, we update all of the groups on the findings, whether they were positive or non-detection. So this middle photo is a collection of isolated eggs that we had from a sample. So this isn't exactly what you would see when you have a spoonful of sediment, but after we've removed the eggs from this sediment and we put them in another petri dish to count, that's what you would see. So all of those spheres that you're seeing, those are all Pacific sand lance eggs. You can see lots of small sand grains attached all around the egg. And if you look closely, you might see little drops on the eggs. Those are indicative of Pacific sand lance. I'll show you another photo coming up here. So these are a few photos that us and our partners have found over the years. On the left, you can see a collection of Pacific sand lance eggs. So like I mentioned, there are multiple sand grains attached all around the egg. That is a characteristic of Pacific sand lance eggs, multiple attachment sites. Now, if we move to the center photo, those are surf smelt eggs. They were provided by Peninsula Stream Society. And the photo on the right is also some surf smelt eggs. And you can see there's a hatched surf smelt that was found in the sample. So that was a really exciting find for them. In that photo on the right, the grid marks that you can see behind it, those are 1 millimeter grid marks. So that will give you a nice idea of the size of those eggs in the sample. Okay, so the last step of our methodology involves a lot of computer time, so data entry and storage. Our team at Mabry compiles all the data sheets, and we complete quality assurance and quality control on them, and we input the information into our internal database. From there, we submit the data twice a year to the Pacific Salmon Foundation's Strait of Georgia Data Center, and that's an open access database that's hosted by UBC. This has been adopted by all of the members of the BC Forage Fish Network in an effort to ensure that all of the data is transparent and available to anyone who may use it. And specifically, we're hoping that's people who will be using it for policy management. Now, the team at the Strait of Georgia Data Center has created really beautiful maps that visually showcase the monitoring efforts and the results. And if you haven't already had a chance or haven't heard about their marine reference guide, I would highly recommend visiting it, because there's much more than just forage fish data on there, but lots of different marine monitoring efforts happening in and around the Salish Sea. So I'll provide a link to that as well at the end. Now, currently, we have found 132 positive samples from 37 different beach stations since we started the project. In particular, it's all Pacific Sandlands eggs for us. The yellow dots on this slide will represent positive sites, and the blue dots are non-detection sites. So you can see sort of in the Nanaimo and northern region, we have quite a number of positives. It's a little bit not as fruitful in the Ladysmith area, but as we go down south, we're finding some more positives there as well. Now, our 2022 to 2023 results, we always really showcase these results based on the winter season, as that's our peak activity time. We sample monthly, though. This isn't just winter sampling for us. We sample every month year round, but our detections have only been occurring from the winter months, November to generally February, but usually end of January. And that's known Pacific Sandland spawning season. We revisit sample sites consistently throughout the year, and it helps us determine where and when Pacific Sandlands and surf smelt are spawning. Resampling will help us identify if there have been any changes to spawning habitat, spawning conditions, or events. And identifying changes in composition or parameters that are collected can influence whether or not forage fish are going to use the beach for spawning. Community knowledge also contributes to the reasoning for revisiting sites. Many sites that we sample at are at the recommendation from our citizen scientists who recall seeing spawning events years ago, or who see the fish in the waters just off of the beach. This past winter, we had 3 egg detections in November, 13 in December, and 9 in January. And when I say egg detections, I don't just mean that we found only 3 eggs. It's that 3 different sampling instances brought up a collection of eggs. So that can be anywhere from 2 individual eggs up to, more than 200 or 300 eggs. It really depends. This year, we didn't have any luck in February. That was similar to last year. And overall, we actually had a decrease of eggs this year. And that was something that most of the network had noticed as well. So it might have been some type of environmental impact. We're not quite sure, but something we are looking into. And this past winter monitoring season, there were 16 beach stations in 8 different regions that were recurring positives. So this means that we had evidence of egg detections in previous years on those beaches, which is always a great thing to see, that they're positives again. The hexagons here represent the regions. The number in parentheses represent the stations that had recurring positives. And I've just put up a few of the notable beach stations in each of those regions. Now, I'm really close to ending. I just have a couple more slides, and I really just want to bring some of the information all together at the end here. So there are some really great newsletters that are put out with regards to forage fish monitoring in BC. Every 2 months, us at Mabry, we release our newsletter, The Forager, and it updates citizen scientists on findings, forage fish facts, upcoming training sessions, and any other additional information that groups want to share. This is one of our methods to keep citizen scientists engaged and informed, and helps us reach the public. If you'd like to be a part, you can always send me a message and I'll include you on the email list. Similar to us, Peninsula Streams releases the Schooler, and Project Watershed also has a monthly newsletter for their whole organization, which showcases updates on their forage fish program and any volunteer efforts that you can register for. Now, with all of this collaborative effort along the BC coast, I want to bring to your attention what our collective work contributes to, such as nearshore education and involvement, evidence-based policy and management modifications, better conservation tactics, so preserving the marine food web's lifeline, really. Now, as I wrap up, I want to mention that our network, the BC Forage Fish Monitoring Network, is having a spring forum. It is happening next week, so really close to this Forage Fish talk here. You can register for the event. It will have many experts at it who deal with forage fish research, both from Canada and from the US. It's a wonderful opportunity to hear about recent research and updates from the BC network as well. I can provide a Zoom link that you can register for once I finish this wrap up. And I just want to say thank you again. Thank you to Wonders of the Salish Sea for having me. Thank you to my partners in this, and thank you to the funders who have been able to make it possible. We're really excited about Forage Fish and where the research is going to go, and always excited to talk about it and bring new people involved. So thank you, everyone.